Hello, welcome to Group Problems 9 by Camille Dowse and Katie Jones, and we are Group K. So number 1, diagram an intermediate stage of DNA replication in the bacterium E. coli. Draw two replication forks proceeding from a single bacterial origin of replication known as the ORIC. It may be easier to draw a linear DNA model rather than a circular DNA model. Indicate the following in your drawing, and I have the list of things that you need. So this is the drawing that I did. So I labeled everything. I added the DNA gyrase, but I also showed the polarity of the strands, all the enzymes, including the helicase, primase, the polymerase subunit alpha, and the DNA ligase. And I also included the linked Okazaki fragments, which are shown kind of as fragments in red.
So for question 2, it's in the blanks provided below, indicate the protein that best fits the description. And for some questions, there can be more than one correct answer. So for A, it's this protein complex synthesizes primers in eukaryotes, and it would be DNA polymerase alpha and DNA primase. For B, it's this protein promotes the separation of AT-rich sequences within the ORIC, and this is DNA A. This subunit of DNA polymerase 3 contains proofreading activity, and this is for part C, it's epsilon and theta. For D, it's this protein synthesizes the leading strand in eukaryotes, and it is DNA polymerase epsilon or DNA polymerase delta. For E, it's this protein displaces DNA polymerase 3 from the chromosome to terminate replication, and it is telomerase. For F, it's this protein has 5' to 3' polymerase and 5' to 3' exonuclease activity, and it is polymerase 1. This protein introduces negative supercoils into the DNA ahead of the replication fork, and this is DNA gyrase. This is for part G. And for part H, it's this protein binds directly to ARS elements, and it is the origin recognition complex. For I, it's this protein prevents DNA strands from annealing during replication, and annealing is basically means reforming the hydrogen bonds. And so this is a single strand binding proteins. For J, it's this enzyme seals Okazaki fragments into a continuous strand of DNA, and it is DNA ligase.
So 3A. Those 5 enzymes, prokaryotic or eukaryotic, that consume ATP during DNA replication. So DNA ligase, DNA primase, DNA gyrase, DNA helicase, and MCM helicase are those. 3B. Those 5 enzymes, prokaryote, both of the cell types, that consume DATP during DNA replication, and these can be found polymerase 1, polymerase 3, polymerase alpha, polymerase epsilon, and polymerase 5. Those 5 enzymes that have 3' to 5' exonuclease activity, and these are telomerase, polymerase 3, epsilon subunits, polymerase 2, and polymerase 5. 3D. Indicate one enzyme involved in replication that uses DNA as a template to synthesize RNA, and this is DNA primase. And 3E. Indicate one enzyme that uses RNA as a template to synthesize DNA, and that's reverse transcriptase.
So for 4, suppose you're trying to synthesize DNA in the lab. In a single-stranded DNA model, 5' ATAGCAGGACC ATTCCTTTAAGCA 3' will serve as the template for the reaction. This DNA template is mixed with the DNA polymerase 3 holoenzyme, the primary sequence. With that following sequence, nucleotides, which are given in parts A through D, and a buffer containing magnesium, which is a cofactor necessary for polymerase activity. You decide to perform 4 DNA synthesis reactions, which are parts A through D. Determine the sequence of the DNA strand synthesized if you use the nucleotides described in parts A through D in your experiment. Assume that if a DNA polymerase cannot incorporate a nucleotide into the nucleic acid strand, the reaction will terminate after the last incorporated nucleotide. So for A, since the nucleotides in this reaction include DATP, DCTP, DGTP, and DTTP. And this is the following DNA strand with the nucleotides. For B, the nucleotides in this reaction include DATP, DCMP, DGTP, and DTTP. And there's the following strand. For C, the nucleotides in this reaction include DATP, DCTP, DGTP, and UTP. And this is the following strand. And then for D, it's the nucleotides in this reaction include DATP, DCTP, GTP, and DTTP. And there's the following strand.
Alright, so 5. So below is the list of proteins involved in DNA replication, and you can see this whole list in front of you. So 5A. Which of the above proteins is found in prokaryotes? And so we have DNA gyrase, TUS, DNA A, DNA polymerase epsilon, DNA primase, DNA polymerase 1, DNA polymerase alpha, and DNA polymerase 3 alpha subunit. 5B. Which of the above proteins is found in the mouse musculus? Sorry, that's Latin. I'm not very good at that. So, oh well, it's a mouse and a mammal, and so it's eukaryotic. So we have DNA gyrase, CDC6, TUS, DNA A, DNA polymerase epsilon, DNA primase, DNA polymerase 1, DNA polymerase 3 alpha subunit, telomerase, and pCNA. 5C. Which of the above proteins in that list is involved in a chromosome compaction, and that is telomerase and TUS. 5D. Which of the above proteins is involved in the initiation stage of DNA replication, and so that's DNA A, CDC6, and MCM helicase. And 5E. Which of the above proteins is involved in the termination stage of DNA replication, and that is TUS and telomerase. And thank you for watching, and have a nice weekend.